have just one functional group. They have more than one. And the basic characteristic of the building blocks of the proteins is that we have molecules, or their building blocks, we call them alpha amino acids. Alpha, and this is the acid group, and this is the amino group, right? So they are the alpha amino acids because the amino group is of the first carbon attached to the carboxylic group. And they have basically, their basic characteristic is that they have these structural characteristics. Next to the carboxylic group, we have the amino group on the alpha carbon. And then the R group here, the rest of the molecule, basically is made out of different groups. Now, we have about 20 amino acids that we need to use to build up the molecules, to build up the proteins. And basically we have 20 different R's here, 20 different combinations. And we have, just starting with the basic things that we know this is a Fisher projection, right? The vertical bond here, the horizontal bond points towards you, the vertical bond points in the back of this plane. The molecules that have the amino group on the right side of the vertical bond are the D amino acids, and the molecules that have the amino group on the left side of the vertical bond are the L amino acids. And if we are going to do the absolute configuration, the R and the S configuration, the L amino acids have the S configuration, and the R and the D amino acids have the R configuration, right? The absolute configuration. And of course, the natural amino acids, they are usually the L amino acids. By saying that doesn't mean that we don't have D amino acids found in nature, but it's very rare. Now, the other thing that we need to realize is that, well, going back to our discussions with the acids and the amines, if we have an acidic group and an amine group, an acidic group and a basic group within the same structure, they are not going to be neutral, right? They cannot be neutral. They are going to have to react. And they react. And basically what we have in what we consider to be the neutral form, this is not the neutral form. What exists is the ionic form, where we have the hydrogen from the acid group has been transferred internally to generate this internal ionic compound. And that internal ionic compound has a specific name. It's called zwitterium, which basically is a German word which basically translates liberally into double ion, basically, right? So we have a double ion. We have a positive ion, ammonium on the amine group, and carboxylate ion on the acidic group. And this is basically just two examples of two of the amino acids that you have. This is alanine, and this is phenylalanine. Phenyl is this one. And the smaller one, this is the simpler amino acid, basically. That's glycine, right? Again, in their natural state, the amino acids are not going to be this. They are not going to have this form. They are going to have the ionic form, where we are going to have an ammonium ion and a carboxylate ion within the same group. Now, another classification of the amino acids, in addition to the L and the D and the S that we had before, another classification of the amino acids is the nature of the R group. If the R group has another carboxylic group or another acidic group, they are going to be the acidic amino acids. And if the R group is having an amino group, they are going to be the basic amino acids. And if the R group doesn't have a carboxylic group or an amino group, then we have the neutral amino acids. Neutral amino acids examples: glycine, which the R group is a hydrogen; alanine, which the R group is a methyl group; or phenylalanine, where the R group is a phenyl methyl group. In the basic amino acids, on the R group, we have additional amino groups basically, right? This is the alpha amino group, right? And on the remaining, on the R group, we have additional amino groups which give the basic character. We have the amine group here, we have this pyrrolidine group, and this for arginine, histidine, and lysine. And the acidic amino acids will basically have the aspartic acid, where you have the carboxylic group attached to the methyl group, and the glutamic acid, where you have two carbons between the amino group and the new carboxylic group. Okay, and these are all the most of the amino acids that we have, and as I said before, we are not looking for new amino acids. We know which they are. They are about 20. They are the ones we are using. About 10 of them we are able to produce in our body, and 10 of them we cannot produce them, and we need to eat them. We need to introduce them as they are into our body. Those are called the essential amino acids, and the ones that we produce in our body, they are not really essential because we can eat something and convert it into those amino acids. The important thing, because as we said, the reason that I'm using the PowerPoint, as I explained to you before, is because I'm kind of lazy, and we need to write very large molecules, right? So, and this is not, I didn't discover that one. This is the reason why we are using symbols of the amino acids. You can see we have the names of the amino acids, alanine for example, and the first three letters of the name, they become the symbol of the amino acid. And also a new way of drawing them, we can also just use the first letter of the amino acid the same, but in some cases we are going to have a conflict. Asparagine here, ASN, cysteine, CYS, glutamine, GLN, and so forth. So for every amino acid, we have basically the first 2 or 3 letters of their name to use them as symbol, and you will understand why and how convenient this is going to be, because we are going to see molecules that have hundreds of amino acids. Proteins, they have hundreds of amino acids, so you cannot really go writing the structures of hundreds of these things one after the other, right? The good thing though is that yes, they may be hundreds of them, but all of them are connected basically within the same bond. Yes?
Do we have to memorize the symbols and the symbols? The names and the symbols?
This is organic chemistry. The answer is no, but if you do biochemistry, the first thing they are going to make you do is memorize them. Okay?
Mm-hmm.
But if you practice a lot and you use them in many exercises, eventually you are going to memorize them whether you like it or not. Okay? So the next thing that we are going to study are the properties of the amino acids. And connecting you back to your general introductory chemistry courses, when we have an acid compound or a base compound, we know that we have dissociation reactions. They transfer protons and they accept protons, right? So we are going to start, we are not going to start from the natural state of the amino acids, right? This is, we said the ionic form is the natural state of the amino acids. Let's start with the hypothetical amino acid in a very strong acidic solution. If we have a very strong acidic solution, for example, like pH 2 or 1, all the possible sides of the molecule that they can accept a proton, they are going to be protonated, right? So we are going to have the protonated form of the organic acid and the protonated form of the amine group, which is going to be the ammonium form. And the reason that the acid group is going to be protonated is because the organic acids are usually weaker acids than the inorganic, than the non-organic acid, right? If the pH here is 2 because of hydrochloric acid, right? And keep that in mind when we talk about the proteins later. So if the pH is generated by hydrochloric acid or any other strong acid, that means the organic acid is going to be protonated. So this is the protonated form of the amino acid, and the important thing that you need to understand here, in the very strong acidic environment, the protonated form of the amino acid is going to be positively charged, right? the overall charge of the amino acid here is positive, right? Because of the ammonium salt. Then, if we increase the pH, right? If we increase the pH, go from pH 2, let's say, to pH 6, the more acidic group of the two protonated groups is going to lose its hydrogen, right? So which is the more acidic group? We have a nitrogen to hydrogen bond and an oxygen to hydrogen bond. Which of the two bonds is more acidic?
The carboxylic.
The carboxylic bond, right? That's the acidic group. So, and the reason is because oxygen is more electronegative than the nitrogen, therefore the oxygen is going to attract electrons stronger than the nitrogen. So, and that form now is the neutral form, right? At pH 6, you have one negative charge and one positive charge. One negative and one positive, they cancel each other out. So this is the neutral form of the amino acid. And if we continue increasing the pH of the environment, we are going to go, let's say, to pH 10, which is an extremely basic pH, and we are going to have all the groups that we have protonated, that we started with the protonated, all of them are going to lose their proton, and we are going to have the free amino group. And again, at a strong basic environment, pH, the organic bases are weaker than the other bases, therefore they are going to lose their proton, their hydrogens faster. So, some other definitions that we know, that we have. Basically, this sequence here is a two dissociation reaction sequence, right? And for each step, we can write the equilibrium constant, right? So when we write the equilibrium constant, we are going to have the concentration of the products above the concentration of the undissociated form of the reactants. And this, for the second step, is going to be the concentration of this above the concentration of the undissociated product. Now, another definition that we need to be aware of, the point where our amino acid, or by extension our peptide or our proteins, are going to be neutral. The number of positive charges are going to be equal to the number of negative charges in the molecule, like here, right? Number of positive equal to the number of negative charges. Here we have excess of positive charges, therefore it's positively charged. Here we have excess of negative charges, therefore it's negatively charged. Here we have equal number of the positive with the negative charges, right? So at the point where the amino acid or the protein or the peptide have the same number of positive and negative charges, that pH is called the isoelectric point. So whether it's an amino acid, a peptide, or a protein, when the total charge of the molecule is zero, is neutral, then we are talking about the isoelectric point. And of course, we have three different kinds of amino acids now. We have the neutral amino acids, we have the acidic amino acids, and the basic amino acids. And because they have different groups, different numbers of groups, they are going to have different pH where they are going to be neutral. Their isoelectric point is going to be different. So for the neutral amino acids, usually the isoelectric point is at pH somewhere between 6 to 7.5. For the acidic amino acids, the neutral their neutral pH, or the pH that they are going to be neutral, is somewhere between 2 and 4, and for the basic amino acids is somewhere between 10 and 12. And we are going to see some applications of the reaction that we had before. This is glycine, the simplest amino acid, the only one who doesn't have a chiral carbon basically. And you can see this is the protonated form, positively charged, dissociation to the neutral form, and hydrogen cation, and then the neutral form can also be dissociated to this one. For each step, we have one equilibrium constant. And if I replace one of these hydrogens with the methyl group, we make the alanine, and we make the same thing. So for the neutral amino acids, we have two-step dissociation reactions and two equilibrium constants. And even also, this is the isoleucine, we can do the same thing and write it the same instructions. For the basic amino acids, for the acidic amino acids, like the glutamic acid or the aspartic acid over here, the protonated form in the very strong acidic environment, we are going to have both carboxylic groups protonated and the amino group as well. So they are going to be a three-step dissociation process. The first step dissociation process, we are going to lose the hydrogen from that carboxylic group, and we are having this anion, this neutral form with the hydrogen cation. For the second step, this neutral form is going to lose the hydrogen from the other hydroxyl group, right? To give us the new hydrogen cation and the dicarboxylate ion now. And the last step in the extreme basic environment, we are going to lose the hydrogen cation from here to give us the negatively charged. And as we said, this is the neutral form. In this case here, we have excess of a negative ion, and in this case, we have two negative ions. So the total charge here is 1 plus, the total charge here is 1 minus, and the total charge here is 2 minus, okay? So you just count the symbols over there. And the same thing for the glutamic acid over here. Plus, 1 plus, 0 charge, 1 minus, 2 minus.
Professor?
Yes.
I don't really understand how it's like going from that to that. Like, I just don't understand it.
This?
Yeah, like how is it dissociating? Like I understand they're losing a hydrogen, however, like
It's
how
a dissociation reaction, you lose the hydrogen from here.
are they adding
It's
anything?
an equilibrium. Oh, well, this one takes place in water. It's a solution.
Okay, got it.
All these things that we do, they are in solution. And we are interested more in the things that take place in water
Is our
because
body?
our cells are basically a water environment.
Mm-hmm.
Okay?
Okay, thank you.
So if we move on to the basic amino acids, the protonated form in the strong acidic environment is going to include the ammonium ions, right, plus the carboxylic group. The first dissociation in water in a solution, oro is, the first dissociation is going to give us 2 positive charges and 1 negative charge, right? So the overall here is 1 positive charge. The second dissociation, which is going to be the removal of the hydrogen cation from this amino group, is going to give us the neutral form, right? Plus 1 minus is 0. And the third dissociation, by removing the hydrogen from the last amino group, is going to give us 1 negative charge. So we have 1 positive, 0, 1 negative. And we can do the same for all the basic amino acids. For example, we can have here the histidine, where we have the protonated form, we go to lose the hydrogen from the carboxylate to give us this, and then we go to lose the hydrogen from the amino group here to give us this, and then we lose the hydrogen from the second amino group to give us the final carboxylate.
Professor,
Yes.
Turn the lights off in front.
Hmm?
The lights in the front. Can you turn them off?
You want them on?
Off.
Oh, they are off. You want me to turn the other
these are
lights?
the front, yeah.
That is kind of difficult. You need to, okay. No. No. No.
Yeah, that's fine.
That's better?
Yeah.
Okay.
Yeah.
So I need to make the note, the first 2. Okay. That is very high technology for me. Okay, people. Then, the next classification, we spoke about the alpha amino acids, how they behave in water, what their charges are, going from very strong acidic environment to very strong basic environment. Then we are going to look at the combination of the amino acids, which we call peptides. And if we have two amino acids joined together, we have dipeptides. If we have three amino acids, we have tripeptides. If we have more than three amino acids, we have them as polypeptides. And basically, we have, when we join the amino acids together, this is the bond that is forming, right? Mm-hmm. And you can see, being technically correct, we have the protonated form of the amino group and the carboxylated form of there, but this one is an amide bond, right? Remember when we spoke about the formation of the amides by heating organic acids with amines and then heating them a lot to dehydrate them? So that's basically what happens. It's an amide bond between the carbon or the carboxylic group and the amino group of the other amino acids. And you can see here in the tripeptide, we number the peptides based on the number of the amino acids they have. If they have two, they are dipeptides. If they have three, they are tripeptides, right? So this is one amino acid, this is the second amino acid, and this is the third amino acid, right? And if we go to the polypeptides, basically, the polypeptide is a polymer where it's a repeating sequence of the amide bonds with the only change as they are. The only difference to be the R group of the different amino acids. If we didn't have a change in that R group, we would have got a polymer like the ones that we said before, where we have a molecule and we have the repeating unit of the molecule. But here we have, because these are natural molecules, the R group is going to be different for every amino acid. And again, we don't really have all possible amino acids in nature, or nature gives us about 20 amino acids, which we are going to be about 20 amino acids that we are finding in all the natural molecules. Now, some other definitions that we need to be aware. In every sequence like that, or like this, there is going to be one amino acid which is going to have the free, the carboxylic group free, and one amino acid which is going to have a free amino group. The one that has the carboxylic group free is called the C-terminal amino acid, and the one that has the amino group free is going to be the N-terminal amino acid. By agreement, by convention, everybody agrees to write the N-terminal amino acid on the left side and the C-terminal amino acid on the right side. It's not really written in stone. You are going to see them written any way possible. The important thing for you to know is that the amino acid that has the carboxylic group free is the C-terminal, and the one that has the amino group free is the N-terminal amino acid. Okay? So the next thing is how do we name them? Right? The IUPAC rules that we learn apply here. Usually, when we are going to name the peptides and the proteins, following the IUPAC rules, we are going to name them all as substituents of the C-terminal amino acid. So in this case, the C-terminal amino acid is a serine, before is the alanine, and here is the phenylalanine, right? So the name is going to be serine. Then we convert the alanine and the phenylalanine into a substituent name. And how do we do that? We change the ending to YL. So this is going to be the alanine serine, phenylalanine serine. Basically, that's the name. Now imagine if this peptide has 50 amino acids. You are going to have to name all the 50 amino acids in the order that they show up in the amino acid, and you are going to end up with the last amino acid as the name. Here, this amino acid here, which is this peptide, which is a peptide, has only 2 amino acids. is going to be the phenylalanine, right? And over here we have all these other
Wait,
two
Robert.
names. Yes.
Where do we get the blue, the alanine? Yeah, where do we get that?
The name of the amino acid is alanine. So you change the ending to YL
How
to
do
be
I?
substituted.
How would I know if I saw the molecule though? Like if I saw the molecule? how
Well,
would I
if you
know?
see the structure of the molecule, you need to be able to recognize that this is the alanine group.
Got it. Okay.
The alanine comes from the alanine amino acid. Now, here we have a peptide which we have 1, 2, 3, 4 amino acids. It's a tetrapeptide, right? And we have these tetrapeptides, we have the names of the tetra, as we said, the names are going to show up of the amino acids in the order that they show up in the structure of the amino acids. So it's going to be the glycine, alanine, proline, serine, right? The end amino acid is serine. In the second one is glycine, alanine, serine, proline. Proline is the last amino acid, and all three of them have the proline. But you can see these are different combinations of them. All the same amino acids, the same 4 amino acids, but in different order, right? So in that different order, remember the definition of the constitutional isomers, same formula, but different bond sequence, right? Different order. So that's exactly the definition of the constitutional isomers. 4 different amino acids arranged in different orders, in different combinations in the name. And the name corresponds to each different sequence. And this is not just an academic exercise just for you to be tortured about it. These amino acids, these different sequences of amino acids have also different properties. That's what makes them unique, right? So it's not just they have similar structure, different bond sequence, they have different properties. For example, we have this over here, and this is what we call the enkephalin neuropeptides. They are they are peptides, pentapeptides, I think. They have 5 amino acids, 1, 2, 3, 4, 5, right? And they are basically the same, right? With the exception of the last amino acid, right? This one has methionine as the last amino acid, this one over here, and this one has leucine as the last amino acid. Any idea what these pentapeptides do?
Are they fat?
We've never heard them before. Okay, these are the natural painkillers. They are the molecules that our body develops when we are in pain, and they go to the pain center and they attach themselves over there. Okay? The difference is that because they are made of the amide bonds, we have enzymes that catalyze, that destroy the amide bonds, so the effect is not permanent. Yes?
Would adrenaline be a natural painkiller?
No.
Okay.
Adrenaline is not a painkiller. Adrenaline is the flight or fight thing.
Okay.
Okay? Then another major class of peptides that we have are what we call hormones. Oxytocin, which is a hormone that regulates pregnancy and birth processes, or vasopressin here, which is a hormone that has to do with blood pressure and stuff. So these are cyclic, okay? They are cyclic peptides, and the ring, you can see the sequence of the different amino acids, and you can see that basically they are the same, right? And even they have the same sulfur to sulfur bridge over here, the disulfide bridge, right? The disulfide bridge that we spoke about. The only difference is the amino acid that is attached to the proline. This is, we have leucine, and here we have arginine, right? And different ways of presenting them, this type of structure or this type of space filling structure. Okay? Now, when you go from the amino acids to the peptides, usually called peptides the molecules that have up to about 50 amino acids. Molecules that have more than 50 amino acids, we try to call them proteins, right? Or because they are very, very large molecules. And again, using the symbols of these molecules, of these amino acids, it makes it easy for us to write them because their chemistry is not really as complicated. They are made of amide bonds and then they have the same structure. So when we go to talk about the peptides and the proteins, we classify them into, in studying them, into having a primary structure, a secondary structure, a tertiary structure, and a quaternary structure. So these are classifications to help us study them. The primary structure is basically the sequence of amino acids that we have. And all the proteins and all the peptides are going to have a primary structure. That means there is going to be the order of the amino acids that are connected to each other to make the molecule. And that order is basically the basis that dictates all the other properties of the proteins or the peptides. Then we have the secondary structure, which it's becoming the shape that they take in space. And we have two particular characteristic secondary structures, the alpha helix and the beta pleated sheet, and we are going to see how they are together. The tertiary structure is the overall shape of the protein or the peptide, a three-dimensional shape that is basically including the alpha helix and the beta pleated sheet in a kind of a three-dimensional structure, whether it's going to be like a globular protein, like a little sphere, or like an elongated sphere, or however it's going to be. And then is the quaternary structures, where we have two or more polypeptides joining together and held together in a complex kind of form. So let's start with the primary structure. As I said, the primary structure is the sequence, is the plain sequence of the amino acids. We have one amino acid here. You can see the amide bonds that are connected, and is basically a repeating of the amide bonds one after the other. We have a typical example when we study the proteins or the peptides is insulin, right? An insulin, this is the chain of insulin, the primary structure of the insulin chain, right? It has all these amino acids. Everybody knows what insulin does. Where is it produced in our body?
Pancreas?
In pancreas. And it regulates the
Sugar.
carbohydrates, the
Oh.
sugar in our body, right? So we have two insulin strands, basically. The insulin is made of two strands, the alpha and the beta strand, and both of them are connected with each other through disulfide bridges. Now, the next structure that we have is the secondary structure is the alpha helix, right? In the alpha helix, you can see how the strands of how the protein and the peptide goes around to form a helix. And in every turn of the helix, we have the amino acids connected, right? Connected from this ring to this ring, and the connection is through hydrogen bonds. The carbon-oxygen double bond from the amide group is connected through space with a hydrogen bond to a hydrogen from an amino group. And for every turn of the helix, we have about 3.6 amino acids. So when the helix is forming, right, when the helix is forming like that, the R groups of the amino acids, remember, the amino acids or the peptide is a sequence of amide bonds, and then the R groups, 20 variations of these R groups that are attached to these helix. The R groups are pointing outwards, pointing outside the helix, okay? Away from the axis. If this is the axis of the helix, the R groups are pointing away from it. And the helix is held together by hydrogen bonds in every turn, connecting the carbon-oxygen double bond away from the in every term. So, the next one, yes,
The R group is CH3, right?
not all of it. CH3, phenyl CH3, 20 different variations. Remember the structures of the amino acids that we had before? Those.
Okay.
Okay. Now, alpha helixes, this is what, this is another presentation, and you can see again, this is the presentations where we have the hydrogen bonds, hydrogen bond, and these are the R groups pointing away from the center of the helix. Another one that we have is another type of secondary structure is the beta pleated sheet. And the beta pleated sheet is basically we have parallel strands of the peptides arranged next to each other, and then each one of these strands is connected to the other one through hydrogen bonds. Okay? You can see the hydrogen bonds connected through the parallel, connecting each parallel strand to the next one. And again, the hydrogen bonds are between the oxygens and then hydrogens attached to the nitrogens, right? So the R groups now, in this case, they are positioned above and below the plane of the strands. And I think in the next slide we are going to see how they are. Okay, when we put in the previous slide, the pleated sheet was giving them a representation that it was a planar, but it's not really a planar. It's kind of a wavy sheet where the angle here, this angle is 109.5 degrees. The angle between the two waves, if you want to put it that way. And 109.5 degrees is the trahedral angle, right? Because this carbon over here, right? The carbon that controls that angle, that is connected to the amino group and the carboxylic group, is a tetrahedral angle. It's a tetrahedral carbon, has CH3 sp3 hybridization. And again, the R groups above and away from the pleated sheet, right? Each one of these has different each one of the two basic structures, the alpha helix and the beta pleated sheet, have different structural properties. The alpha helix gives flexibility to the protein strand, and the beta pleated sheet provides strength for the protein. Now the combination of both of them, we are going to have in the protein, we can have the beta pleated sheet and the alpha helix together. It doesn't mean that one protein is going to have only alpha helix and the other is going to have only beta pleated sheets. They are a combination of them, and they are parts of the proteins that do not really fall into this classification. So that takes us to the next structure, where we have the three-dimensional structure. And as we said before, we have our basic example is the insulin, where we have two chains, the alpha chain, the blue chain, and the beta chain, which is the red chain. And both of them, and you can see they are connected to each other with disulfide bridges. When we change the disulfide bridges, when we break the disulfide bridges, basically the two strands are going to be separated. But when insulin is together, then it's going to have both the disulfide bridges, and essentially we have alpha helixes and beta pleated sheets as part of the proteins in the three-dimensional shape. Now, when we go to this three-dimensional shape, the tertiary structure of the proteins, logic, right? Where do all these proteins exist in our cells? Our cell is a water environment, right? It's a solution of molecules in water. Therefore, when a molecule like that forms on the outside, on the surface of the protein, right? The surface of the protein, what are we going to have? Hydrophilic or hydrophobic groups?
Hydrophilic.
They're going to be the hydrophilic groups, right? Because we need them to interact with water. That's how they become soluble in water. And inside the protein, inside these loops and everything, we are going to have the hydrophobic groups, the lipophilic groups, right? Where they are going to be away from the water, so they are not going to minimize the interactions with water. So inside the protein, we have maximizing the interactions, the lipophilic interactions, and in the surface of the protein, we have maximization of the hydrophilic interactions, the interactions with water. So, what are these forces that we have? We have hydrogen bonds and polar interactions, carboxylate ions, ammonium ions, groups that can make hydrogen bonds with water are on the outside. Inside of the proteins, we have usually the non-polar interactions, failure to failure groups interactions. We can also have ionic interactions and hydrogen bond interactions at key positions to hold the protein together. And also we can have, the extreme case is when we have covalent bonds like the disulfide bridges. Sulfur is generally symbolized in yellow color. So these are the disulfide bridges of the two insulin strands. Okay, so we have the primary sequence of the amino acids, helices and beta pleated sheets as the main characteristic of the secondary structures. The tertiary structures is when the helices and the beta plated sheets, they join together to form a three-dimensional structure where at the surface of the protein we have the interactions with water molecules through hydrogen bonds and polar and ionic positions and ionic groups. And the inside we have, again, the non-polar interactions, but also sometimes polar and hydrogen bond interactions to hold the protein together. And in many cases, like in the case of the insulin here, we have the disulfide bridges. Now, the last one is the quaternary, and the quaternary structure is very important even for insulin is important, but it's important for our blood, for example. In the quaternary structure now, we take three-dimensional structures of the proteins and we bring them together to form aggregates where for insulin, for example, we have a dimer, of it, or a hexamer of it, which the hexamer is essentially three dimer units together, right? And that's basically the quaternary structure, a complex structure that is generated by combining individual tertiary structures. In our blood, the heme units, in our hemoglobin, we have heme units, which each unit has each strand has a heme unit inside. These are the green areas, and we are going to see the structure. And we have two beta subunits and two alpha subunits. The alpha subunits are the red ones, the beta subunits are the blue ones. And this is the quaternary structure of our hemoglobin, which carries basically through the heme units, carries the oxygen to our cells and removes the carbon dioxide from our cells. Wow. Similar structures we have for in our the myoglobin has the porphyrin unit over here, and that one is usually found in our heart muscles. That's why our heart muscles are very they are red basically. And the porphyrin, this unit where we have the iron there, is the unit that takes that captures the oxygen and takes it to our blood, right, and holds it for us. So when we have poisoning, when iron captures the oxygen from the atmosphere through our lungs, carries it to our cells, right? Then, if we have something that can replace the oxygen, like carbon monoxide, for example. This is a complex molecule. If we replace the oxygen with carbon monoxide, the complex with carbon monoxide is about 200 times more stable than the oxygen. Therefore, this one is not going to be able to combine with oxygen anymore. It's not going to be able to carry oxygen to our muscles. So the person who is with carbon monoxide poison, suffers with carbon monoxide poisoning, is not going to be getting enough oxygen, and essentially is going to die from deprivation of oxygen, right? So usually people who suffer with carbon monoxide poisoning, they are put in chambers where there is increased pressure of oxygen to force the oxygen into their myoglobin. Okay, now chemistry. Now after we learn everything that we could about proteins, let's turn our attention to chemistry. As I said before, the chemistry for these molecules is not really a complex chemistry. They are reactions that we already know, and it's basically applied chemistry. So if we are dealing with peptide bonds, right? So in other words, we learned that the amide bonds before we can hydrolyze them. So if I take a peptide like that and hydrolyze it, that means hydrolyze it, heat it with sulfuric acid and water, all the amide bonds are going to break, right? And since we are having acidic conditions, we are going to get the ionic forms of the molecules, right? In the strong acidic conditions, we should be getting the the protonated amino groups, right? And in this case, we should be also having the oxygens, instead of having a negative charge, they should be having a hydrogen, the protonated form. But usually when we do these reactions, we try to isolate those. So I just show the final product here, not the immediate product of this reaction. But technically, the oxygens, all the oxygens should have a hydrogen as an attachment to them. Okay, again, this is a tripeptide, right? One amide bond here, the other over here. If I heat them with sulfuric acid and water, that's the typical conditions basically. We are going to make the three final products. We adjust the pH at the end of the reaction to isolate these, to precipitate them out basically. Or if instead of writing the structures, we can write we can write the name of the tripeptide, hydrolyze it, and break it into the three amino acids as before. Now I hope you had breakfast before you come here, right? So we spoke about the proteins having a particular structure, and that structure of the proteins is controlled by the different forces that operate in the protein, like hydrophilic forces, like hydrogen bonds, polar interactions, and hydrophobic forces like the London forces, non-polar interactions within different parts of the molecules. Now, and those forces are controlled basically when we have them in solution. They are controlled by the pH, right? By the different conditions that we have. So if we change these forces, then the protein loses its structure. It's not going to be, it's not going to have the structure that we have. So if we don't have the structure, if those conditions change, then we are going to have what we call the change of the structure of the protein. The change from the natural structure of the protein is what we call denaturation of the protein. So we can have changes in heat, in temperature. We can have changes in the solvent. We can have changes in the pH that are going to change the structure of the protein. A typical example is the eggs. This is the egg, where you have the egg in its natural state, the yellow liquid, the yolk in the middle. If you fry the egg, you change the temperature, and you fry the egg, you get the white the protein, right? The yellow protein becomes this white solid material, or if you boil here again, the yellow liquid becomes a white solid, and then the yolk becomes also a solid. So we have changes, this is the typical changes in the temperature. Another example of a protein denaturation is the changes that are happening in the milk. If we milk, when the milk gets sour, that's how we make cheese basically. We change the pH in the milk. When we change the pH in the milk, essentially the proteins of the milk, they join together and they form curds, and this is the liquid that they were suspended into. So that's basically what happens, and this is the natural stage of the protein. When we change the conditions, the protein basically unravels to give us a different structure. Okay. A basic class of proteins are the enzymes, right? We've been talking about the enzymes over and over again. And enzymes are essentially proteins which are basically tiny biochemical reactors in our body. They are the molecules who can make all these reactions that we have been studying here. They can make them happen faster and more efficiently in our cells, and they usually can increase the reaction rates by 1 million to 1 trillion times faster. They are very specific. They operate, each one of the enzymes, they have a specific substrate, a specific molecules that they operate on. And many times the enzymes, they need, in order for them to react, they need to have what we call a cofactor or a coenzyme, which that cofactor or coenzyme can be a metal cation or can be another molecule that they can join together to work. So we name the enzymes. There are about 6 categories of enzymes, and we name them to indicate what they do and the substrate they are operating on. For example, here we have hydrogen peroxide with a catalase enzyme becomes water and oxygen, right? It's catalyzed. We are having a catalysis. Here we have the tripeptide phenylalanine serine with the carboxypeptidase is cleaving the amide bond between the alanine and serine. Remember, all these amide bonds that we have in the proteins, each one of them has a specific peptide to cleave them, right? Not all of them. So some of them are specialized in cleaving the C-terminal amide bonds, some of them the N-terminal bonds, some of them only bonds between phenylalanine and serine, and so forth. Okay? So, there are classifications, some of the classifications. Oxidoreductase enzymes, they catalyze oxidation, they catalyze reduction reactions in our body. For example, we have this ketocarboxylate molecule over here with an oxidoreductase. The carbon-oxygen double bond becomes carbon-oxygen single bond. That's a reduction reaction, right? If we have a class of enzymes that we call transferase enzymes, which means they can transfer one group from one molecule to the other. You can see here, we have the amino acid over here, alanine. We have the glutamic acid over here with a transaminase enzyme. That means an enzyme that transfers an amino group from one molecule to the other. The amino group from the alanine is transferred to the glutamate area. So the alanine becomes the keto carboxylate, and the dicarboxylic acid here becomes the glutamic acid. So moving an amino group from one molecule to the other. Hydrolase, as the name says, they are molecules that are hydrolyzing functional groups. We have the lipase enzymes, which basically they hydrolyze the ester groups in the lipids. So with the lipase, the triglyceride is going to become the triol and is going to release the fatty acids. Isomerase groups, isomerase enzymes, they catalyze the isomerization of molecules. And you can see here, this is the phosphate ester, keto group, hydroxyl group, isomerase enzyme, the phosphate, is converting this carboxyl double bond to a hydroxyl group and moves the carboxyl double bond to the first carbon. Okay? The lyase enzymes, usually, you can see what here we have a double bond, which we have an addition of the double bond of water. We have the fumarase enzyme, because this is the fumaric acid, and the water is added across the double bond to give us the hydroxy compound. and ligase enzymes, they catalyze the bond formation. Example, this is the typical ATP and ADP situation where we have the pyruvate carboxylase using ATP is going to connect this carboxylate ion with the carbon oxygen double with the carbon dioxide to give us, to add another carbon to the chain, right? To add the carbon oxygen double bond over there, make the diacid, and we are going to release, the reaction is using energy released from ATP, which the ATP is converted to ADP and releases the phosphate ion in the process. Now, just a few things on how the enzymes work. I mean, as we said before, the enzymes are catalysts. They make biochemical reactions, and they are very specific in what they do. They are very specific in the reaction they catalyze, and very specific on the substrate that they are going to operate on. And people compare that activity into a lock and key situation. That means for every lock there is a particular key that is going to open it, and that was the original understanding of how the enzymes work. In other words, from the lock and key you can see this is the enzyme, this is the substrate goes into the enzyme, fits perfectly, and you have the complex between the enzyme and the substrate, and then when the reaction takes place, the complex is released. Again, the same thing over here. This is the enzyme, these two fit perfectly into the enzyme, and that part of the enzyme where the substrate fits perfectly is called the active side of the enzyme. After the reaction takes place, the substrate is released as the product. Now, there is also, as our understanding of the mechanisms of the enzyme, of how the enzymes work, we came to realize that there are some enzymes that their active sites do not necessarily have to fit the substrate. They may be different from the shape of the substrate, but that changes during the reaction. When the substrate and the enzyme initiate their interaction, we have the enzyme active site adjusts itself to accommodate the shape of the substrate. Mm-hmm. And that is called the induced fit model, where during the interaction we have an adjustment to the enzyme to accommodate the shape of the incoming substrate. Then the enzyme, the complex is forming, the enzyme with the substrate, and then the product is released after the reaction is completed. So, when do the enzymes work best? Or what kind of conditions? As proteins, they have to have optimum conditions for them to react, right? So the optimal conditions for the enzymes to react are the natural temperature of our body, right? The natural temperature of our body is 37 degrees Celsius, right? So that's the temperature that all the enzymes are designed to work. If the temperature goes over 50 degrees, usually they suffer denaturation, their shapes change, and they are not active anymore. I don't think that any one of us survives to a 50 degrees Celsius, right? It's not good. By that time, the denaturation of your enzymes is going to be the least of your problems. And basically, also there is an optimum pH that the enzymes work. It's going an optimum pH, an optimum ion concentration that they work, and so forth. For most of the enzymes, the pH that we have that they work is the pH of our cells, which is about 7 to 7.4, right? That's our blood pH and so forth. But there is a particular group of enzymes in our body, the enzymes that we find in our GI system, in our gastrointestinal system, and those enzymes, they can work at very, very strong, very, very low pH, because the pH of our stomach. Anybody knows what the pH of our stomach is?
Very acidic.
It's 3, which is basically a 10% concentration of hydrochloric acid, right? So,
Professor?
yes.
So, is there like a real life example of how they accommodate the?
Say it again?
Is there like a real life well, of course, but what's a real life example of the enzyme accommodating to the shape? No, I don't even know how to explain it.
Pay attention here. You
Okay.
will get your answers. Okay, so when do we have, when we realize, when we understand the mechanism that the enzymes operate, and then we realize that many of these enzymes control situations of health, right? So they're changing the mechanism or changing the nature of the enzymes has adverse health conditions. So we should be able or develop medicines that interact with enzymes to cure diseases. So we have what we call the allosteric control, which we can have positive allosteric control and negative allosteric control. Remember when we said the induced feed, the enzyme has an active site where the substrate is going to go and make a connection to make the complex. Well, we can have an inhibitor for the enzyme where the inhibitor doesn't necessarily interact with the active site of the enzyme, interacts with another part of the enzyme. And by this interaction, it changes the shape of the active site of the enzyme, and then the substrate cannot connect, cannot interact with that. Therefore, it's preventing the substrate from interacting with the enzyme, but it's preventing it by indirectly in another part of the enzyme operating, right? And that is a negative thing. That is an inhibition of the activity of the enzyme. On the other hand, we have, it's like a cofactor here, we have a situation where the activator is going to come, interact with the enzyme, modifies the passive potential active site of the enzyme, modifies it to become the right shape to interact with the substrate. In this case, the molecule that is interacting with the enzyme, it becomes the activator. It activates the enzyme to become more reactive. So we can have the positive, increase of the reactivity, and the negative, then decrease of the reactivity of the enzyme. We have enzyme inhibitors, which they are molecules that can go into the active site of the enzyme. They can bind temporarily, and then they can be released, or they can be connected, interact permanently, and then they make the enzyme unreactive. The reversible inhibitors, they are basically competing with the substrate of the enzyme, and the reversible inhibitors, they are permanently attached to the enzyme, making it permanently unreactive. So, both of these inhibitors are essentially competing with the substrate. And here is an example so you get to understand it better. So here we have the enzyme, right? It's a protein, this is the shape of the protein, this is the active site of the enzyme, and this is the succinate. succinate, we have carboxylate here, and the succinate goes into the active site of the enzyme. This is how it goes in. We have an interaction with the carboxylic group here and the carboxylic group here. And at the end, when the reaction is over, we are going to generate a double bond over here, right? The carbon-carbon double bond. You can see it over there. And this is the reaction that is going to happen. The succinate becomes the fumarate, the carbon-carbon double bond. Now, if I take a malonate, which is the same dicarboxylic acid, but instead of having two carbons between the carboxylic groups, it has only one carbon. It goes in, right? It fits into the active site of the enzyme, but there is no reaction taking place because it doesn't have the two carbon to make the double bond. And because this one now is in the active site of the enzyme, it's going to prevent the succinic acid from entering the enzyme and becoming the fumaric acid. So this one is an inhibitor that prevents the actual operation of the enzyme. Same thing with penicillin, right? I'm sure you know what penicillin is, and you have an idea what penicillin does. Penicillin essentially prevents or destroys the microorganisms for forming their membranes. And here is how it does it. Right? It enters into the membrane synthesis and breaks the membranes apart. The chemistry of this is that in our there is a glycopeptide in the membrane system, and and penicillin, or here you can see it better. This is the active site of the penicillin, and this four-member ring here is a lactam, right? So it's a lactam, and because it's a four-member ring, it means it's a high-energy structure in the molecule. It means it's a very reactive structure. So under nucleophilic attack from the hydroxyl group, that's a serine residue. After a nucleophilic attack from here, it forms the ester, right? And when this active site of the enzyme interacts with penicillin to make this structure over here, it prevents it from forming the membranes of the microorganisms, and then the cells of the microorganisms, they are destroyed. Their membranes are opening and they die. And that's basically the antibiotic activity of the penicillin, and that's the miracle drug, the miracle antibiotic that was discovered in the 30s, basically, and ever since it's being used as antibiotic. Now, the other thing that you need to know, we have many antibiotics, but these microorganisms are adjusting to those antibiotics very, very fast, and essentially they become used to them, and they develop their own defense mechanisms, and many of the antibiotics become less effective, so there is a constant need to develop new antibiotics adjusting to the new generations of microorganisms. Yes?
Like flu shots, right? They always make different.
Flu shots are inducing the formation of antibodies, not enzymes.
Oh.
Okay? Another example is the example of these catalytic mechanisms, where we have similar molecules, right, that can take their place into the enzyme. In this case, we have the sulfonamide, which can react, can take the place of the amino benzoic acid. And in the if we take it, or if we add the sulfonamide, it's going to interact with the enzyme. The reaction is not going to go any further, and this is in the process of the formation of the folic acid. Or if we do not use this one, the amino benzoic acid is going to interact with the enzyme, and then we can bring this molecule to stop the process of the formation. Or here is the regular process. The amino benzoic acid interacts, generates this one, and goes through the enzyme to go all the way to the nucleosides. And again, we can make the last thing that we have what we call the proenzymes, which the technical term is zymogens. In other words, they are molecular structures that we have in our body, and under the right conditions, they become enzymes. Examples is the trypsinogen of the enzyme trypsin. These are our gastrointestinal enzymes. That they are the enzymes that break the larger molecules into smaller molecules for us to be able to absorb it. And essentially, in the trypsinogen, we have it. The molecule interacts with enterokinase, and then the trypsinogen is activated and it becomes the activated enzyme trypsin that is going to go through the process. Trypsin at the end is going to go and hydrolyze the large molecules, proteins, carbohydrates in our body to be able to absorb them. And also here we have a situation where you may have heard ACE inhibitors, right? We have some of the inhibitors, they block the conversion of angiotensinogen into angiotensin. In other words, if we know the precursor of the enzyme and we know what the enzyme does, in this case the angiotensin increases the blood pressure, and if we want to control the blood pressure of sample of the patient, we try to prevent the angiotensin from being converted by an enzyme, the angiotensinogen from being converted by an enzyme to the angiotensin. The angiotensin, basically, this one is going to be converted to the enzyme by taking away these two amino acids. And we do that by using captopril to prevent this formation. And by that one, by preventing the formation of angiotensin, we prevent the increase in the high blood pressure. And similar ones we have for the ACE inhibitors, where we can put the ACE inhibitors. Essentially, they deactivate the enzyme, the HIV protease, which promotes the formation of the AIDS virus. Okay, I think this is the last one, right? Yes, that's the last one. Okay, people.